With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, have a look at the question. Question is Genomic DNA library means options given are a collection of literature about DNA, a collection of organisms for extracting DNA, a collection of total genomic DNA of a single organisms, a collection of gene vector. So if we'll see the genomic DNA library, these days these genomic DNA library are widely used for studying the complete sequence of DNA. It is being widely used in various experiments because it is a library which contain the genomic DNA. It contains genomic DNA of organisms. Genomic DNA of one organism. It contains all the genomic DNA of one organisms belonging to one organisms. This genomic DNA, it has been stored. This genomic DNA, it has been stored by insertion into specific vectors. It has been stored in specific vectors. Specific vectors are it could be anything, plasmid, cosmid. Usually here are vectors used are which can carry large amount of DNA, large amount of inserts. So these are stored in specific vectors, and whenever these genomic DNA and any sequence is required, it has to they have to introduce into the host cell to multiply and to get the product or desired gene of interest from that library. So in this way, this library has been maintained. Genomic DNA has been isolated from organisms by restriction endonucleases, that particular organism to which genomic DNA library has to be made. Then it has a single insert has been inserted into single cell of the vector, and then these vectors are multiplied into the host cells to get the multiple copies, and at the time of need they can be extracted out, they can be expressed again in the host cell. So in this way it has to be made. That is why it is known as the genomic DNA library. Library means collection, and genomic DNA of a collection of genomic DNA, and that particular of a single organisms. Different organisms have a different genomic DNA, and that's why their libraries are also different. Here a statement says genomic DNA library means collection of literature about DNA. It is not about literature. This is very incorrect statement. Collection of organisms for extracting DNA. This is incorrect. It is simply DNA isolation from the different organisms. A collection of total genomic DNA of a single organisms. This is very correct. I am again and again emphasizing on the fact genomic DNA library, it is specific for a single organisms. Different organisms have a different genomic DNA. Although majority of their DNA is same, but still there are 0.1%, 0.01% chances of having differences. So each and every individual, specifically when they are making the library, so that is of a single organism. So this is correct. Collection of gene vector. Usually students get confused with between these two options. Collection of gene vector is very incorrect because here gene vector are used for storing purpose only. That DNA insert is stored in these specific vectors. So these are not collection of vector. This is a collection of genomic DNA. So this is also incorrect. Option C is correct. Hope you get it. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.